see if it will work here.
John, we already share your screen. Oh, I see.
Okay. Okay, does that look okay?
Yes, Observe, we can see your screen.
Okay, excellent. And you're seeing me switch?
Yes, it is.
Okay, thank you. Okay, so again, I wanted to just say thank you so much to the university, my esteemed colleagues. Thank you. I'm thankful to be together with the other presenters today. And I also am very happy to have both faculty and students present today for the presentation. So thank you for your invitation and thank you for your attention today. So I wanted to start my presentation with mentioning a program that I came to Indonesia under, which is the Fulbright program. So the Fulbright program, I was a Fulbright scholar from February to August in Kupang at Undana in Kupang. And I was able to work with colleagues there in Kupang at Undana, and I was able to make a lot of really important and fruitful connections, both in Kupang, but also across Sumba. And so I really appreciated being able to be housed within Indonesian University during that time. And the Fulbright program is an exchange that goes both directions. So American scholars and students come to Indonesia, but also Indonesian students and scholars can come to America. And so the funding on that program right now, I would say, is back and forth. Sometimes there's more funding, sometimes there's not as much. But the Indonesian organization that administers that program is called Aminef. So Aminef helps administer the program. So I encourage you to, if you're interested in maybe coming to the U.S. as either a scholar or a student, to look up the Fulbright program and see if there's an actively funded opportunity for you to come across. I know that my time in Indonesia was very valuable for me, and I have really appreciated when Indonesian scholars and students have come over to the US, and the exchange is really fruitful and I know really informative for me. So aside from that, if the primary program that if you're interested in postgraduate work, where I am at Goshen College, is a master's in environmental education. So if you really like environmental work and you want to learn more about how to teach environmental concepts, maybe it might be in a classroom setting or it might be at an interpretive center or a national park. If you're interested in sort of learning how to communicate environmental issues, the master's program might be interesting to you. So I just wanted to provide those a couple opportunities there. If anyone's interested, you'd be welcome to contact me. So today I'm going to be talking about old technology, technology that has been common across Earth for a long time and is still some of the most common technology when it comes to managing land around the world, and that's grazing and fire. So, grazing and fire are very, very common in managing grassland and savanna ecosystems all across the world. And so today I'm going to talk about how we're trying to understand how both grazing and fire, how they interact in order to sequester carbon or store carbon in soils and subsoils in these systems. So today I'm going to mention a bit about a multinational collaborative study. So we're already working in the US, which is the bottom picture, and in Kenya, which is the top picture. And I'm interested in working in grasslands in Indonesia. I've been starting to think about grasslands on Sumba. And so understanding what drives carbon accumulation in these grassland ecosystems is becoming really important, especially when it comes to how much carbon these grasslands can store. So when thinking about these two different study areas, right, there's some important differences. And so when thinking about the US, especially Indiana, the state that I'm located in, much of our grassland prairies been have already been destroyed, turned into farmland. And so there's about 6,000 years of human fire interaction in these prairies, give or take, right? So there's been a long historical evolution of grazers on the landscape, but we've lost a lot of carbon from these soils because they've been turned into agriculture. The soils have been disturbed and they've been exposed to oxygen. And so we've lost a lot of our carbon from our soils. Whereas the Kenyan systems that we're working in are very different, right? 1.5 million years of human fire interaction and this long history of grazers, right? But in Kenya, a lot of the native grasslands are still intact. They have not been farmed. And so instead of in Indiana in the US, we're trying to restore some of these grasslands, bring them back to the landscape. In Kenya, it's this idea of protecting the ones from being converted that already exist, right? So very different context in a lot of ways. And so land conversion has been the big issue that releases carbon to the atmosphere, whereas in Kenya, droughts can really be instrumental in releasing carbon to the atmosphere. So when thinking about the ecological underpinnings, right, of carbon dynamics in these systems, right, we know that planetary warming is caused by carbon dioxide emissions, right, from human activity, but also from natural areas and habitat change, right? And when we lose these native grasslands, especially like we have lost them in the United States, lots of carbon has been released from the soil up into the atmosphere. And so, especially in these systems where we've lost so much carbon from our soils already, they have the potential to store massive amounts of carbon. So if we can change the direction that that carbon is going, move carbon from the atmosphere back into the soils, it can be a significant sink, right? And we know because of how common fire and grazing are across the landscape and because of how significant they are in carbon and nitrogen cycling, we know that fire and grazing are likely to play a strong role in how much carbon is being able to be sequestered. So, and I'll just move through some of these quickly. So, right, this idea that carbon is released uh, due to habitat change, but also the amount of carbon released depends on not only the type of habitat, but the types of change happening across that landscape. And this is a piece that is really important for where I live here in Indiana, is historically we have taken these carbon-rich grassland soils and converted them to agriculture. And this has led to possibly, a huge amount of carbon emissions, right? And in the center photo, that's my father and me as a baby on the back of my father. And then the picture to the right is my family now today. So I've had an interesting personal experience with these grasslands. So in the black and white photo in the bottom, that's a picture of laying drainage tile into these prairie areas in order to drain the prairies. And my grandfather was involved in draining the very last prairies, the very last grasslands in the area where I lived. And then in the photo above that black and white, you can see the house. That's the house where I grew up. That was my childhood home. And so you can see all of that would have been grassland, and now all of that is agricultural fields, right? So I grew up, I'm the grandson of someone who finished um, finished draining these prairies and converting them to agriculture. And then I lived and farmed on these old prairie soils, these old that used to be grasslands. And now I'm working with me and my family, my wife, my children. We're now doing work to restore some of these grasslands. So it's been a big circle personally for me to sort of come back to these systems and start restoring them on the landscape. So we can think of plants as a carbon pump, right? In that they're taking carbon from the atmosphere, fixing it through photosynthesis to both form biomass above ground and below ground, but then also to feed the microbial communities that live around the roots in the rhizosphere. So the more productive these plants are, the more carbon, right, we can push into the below ground area, and the more soil organic matter, which is going to be made up of carbon, nitrogen, phosphorus, etc., right, that we can accumulate and make stable, right? And so, like the larger context of the work that I'm doing here in the US, this green area is the entire area where tall grassland, prairie used to be, right? And now almost all of it's gone. 95 to 98% of this grassland is gone. So that's to demonstrate just a bit at how drastic of a landscape change it's been. And so this is the state that I live in. And so I live in this area here where there's isolated prairies. There's more on this side of the state, but there are isolated prairies here on this side of the state too. I live closest to this one. So in the more work that's been done and the more that people look, the more it's becoming clear that grasslands have the potential to store massive amounts of carbon. So much so that they can actually store more carbon than they have the potential to store more carbon than forests, right? A lot of people think about forests because you can see all that carbon above the ground, but grasslands store carbon below ground. And not only in the microbial biomass, not only in the grass and plant debris, but also in charcoal, right? Because these grasslands burn regularly, you can end up with charcoal, which we call pyrogenic. So, and then I mentioned this before, this fact that fire and grazing are so common right across the landscape that they're worth really understanding how they affect carbon nitrogen dynamics. Especially so when you consider how deep some of the roots go in these prairie plants and these grassland plants, right? That there's so much going on below ground that we need to be able to quantify. So one of the central paradigms that we've been testing with some of this work is understanding how in these grazed and burned systems, how exactly carbon is getting into the soil to begin with, right? So is it more to do with leaf litter, right? That you've got plants, dead decaying plants dying from the top, and that as these break down on the surface, that's the main mechanism through which organic carbon is making it into the soil? Or could it be this microbial paradigm, right? Where what's happening below ground with roots and root exudates and microbial growth, that is actually one of the main mechanisms that carbon gets it gets put in place below these grasslands in the soils is through microbes, basically mediated through microbes. So, and here these diagrams are demonstrating the two pathways. So this diagram here, just kind of taking a look at how the surface accumulation might work, right? Mainly through leaf litter getting processed, pushed down into the soil. Whereas this orange pathway over here is, again, largely mediated through bacteria and fungi, where these are the main players. And then necromass, which is just dead microbes, would be the main pathway through which some of this organic carbon would be emplaced and associated with minerals and become stable, right? And then it should be noted that pyrogenic carbon, Pi-C, which is the charcoal left over from a burn, that's always going to be stable, right? That's a very stable form of carbon. So we're interested in this other piece, this idea of how do we get carbon taken out of the atmosphere and then stabilized in these grassland systems. So we've looked at the existing literature, and we can say that results have been uneven, right? There's no clear understanding, and no studies that we are aware of have manipulated both burning and grazing at the same time. So we decided to do that. And sorry, can someone tell me how many more minutes they'd like me to go?
10 minutes again, Professor.
Okay,
10
10 more
minutes.
minutes. Sorry, did you say 10 minutes?
Yes, Professor, 10 minutes.
Okay. So this is our experimental setup. So here's we've got this set up at two farms, same type of setup, in that we have an area where we're grazing and an area where we're not grazing, and then an area where we're burning every third year and an area where we're burning every year. So two different levels of fire and then just one level of grazing. And then we're repeating that 7 times. And we've done this at two farms, a clay-rich farm called Goodrich and a sandy farm called Little Bluestem. So this is just looking at some of the, we'd had to do, because remember, we had to plant these prairies. So this is just a look at some of the steps of prairie establishment over time. And here's a look at some of the coworkers, the colleagues we have, both students and cattle. And this is a look at some of the burning, right, that we've been doing. So this is actually setting up the treatments. And we've started this, really we've started it in the US here, starting in 2016, 2017. That's when we started really planting these two prairies. And we've been doing annual sampling ever since. And then in Kenya, the situation's a little more complicated, and I won't go over this one as in depth, but we have 3 different blocks, right? These are 3 reps. So block 1, block 2, block 3, these are replicates of each other. And these are grazing treatments. So this is letting in mega herbivores and medium herbivores. This is mega herbivores, medium, and cows. This is medium. So when I say medium, I mean like antelopes. When I say mega, I mean like elephant, buffalo, and a giraffe. So this is just medium. This is just cows. This is medium and cows. And this is zero. This is no grazing. So they're actually large electric fences set up to maintain these treatments. And then our burn treatments are around the outsides, right? So we have our control plots. Then we have our low burn intensity and our high burn intensity. So it's a different setup, but again, testing the same idea. And, again, doing controlled burns, just like we showed you in the US. So at the end of the day, what we want to understand below ground net primary productivity. So we need to be able to measure roots. Above ground net primary productivity. So we need to be able to measure the above ground plants. Soil characteristics, carbon, nitrogen, soil organic matter, microbial diversity, microbial biomass, that necromass that we talked about, and then also the diversity of plants, right? So just very quickly, we bury clear tubes to actually take photos of the root systems, and we measure above ground net primary productivity by actually we just cut quadrats, right? And then we collect soil samples. We have been collecting 15-centimeter soil samples from the top for a number of years now, and now we've been selecting deeper soil samples from 0.5 meter deep because we're getting interested in this microbial necromass question. Right, and when it comes to plant composition, this is something called a pin frame, and it has a bunch of rods coming down, and you're able to just quantify forbs versus grasses to understand how that split has changed over time. So I'm not going to talk about a lot of our results. I'm just going to highlight, this is the main one I want to highlight today. And so we're looking at the surface soils here, and we're looking at the deeper soils, the 0.5 meter deep soils over here, right? So the surface soils, they seem to be sort of more changeable. There's not a real discernible signal from the surface soils, but from the deeper soils, what we see is if you burn, right, then reliably, so if you burn every 3rd year, so if you burn every year, you just end up with low carbon, right, because it's too much burning. If you burn every 3rd year, when you don't graze, so this is Goodrich right here, results from Goodrich, and this is results from Little Blue Stem Farm. So we find that if we do graze, the combination of burning and grazing together actually drives down carbon. But then that relationship flips when you don't burn, right? So if you don't burn and you graze, then you actually get quite a bit more carbon than in any other treatment when you graze, but you don't burn. And that's even more carbon than the ungrazed, right? So what this tells us is if you are not burning or not burning very frequently, and you're able to graze, make sure that you've got animals out on the land, then it's likely that you can actually drive up carbon by having cattle remove that low quality material on the surface, and then through urine and feces provide really easily used nutrients to the soils that then trickle down into those microbes, right? Those microbial communities, and it really makes those microbial communities really productive, right? So what we're seeing here, we think, and we've got more data that we're going to be collecting to take a look at this. So what we think we're seeing here is an acceleration of the below ground carbon sequestration, especially at 0.5 meter deep, by stimulating microbes and not from litter from the top. And we're seeing that beat litter from the top. So I'm gonna breeze through a bunch of data here that I'm not going to talk about right now, but folks are free to ask me, additional questions on. So we've got a lot more data and data from Kenya as well, but we're still looking to get our deep soil data from Kenya. We haven't analyzed that yet. So what I wanted to end on is I don't know if any of you have seen the Amarasi system in Timor, but it's this really fascinating grazing system in Indonesia where there's leguminous trees, so nitrogen fixing trees that are cut and brought into the cattle, right? In order to provide forage. And this is an approach that is resilient against drought, right? So especially in areas like Timor and Sumba, where the dry season is really long, it's a system that allows forage to be available more, right? More days of the year. And of course, the tree, right? The tree that's used in this forage system is not native to Indonesia. It's native to South America. And so what you're seeing here is an entire hillside filled with this nitrogen fixing tree, right? And so areas that might have been different types of forest or savanna are now full of these nitrogen fixing trees. So one of my questions that I'd be interested in collaborating on is thinking about how comparing nitrogen fixation rates in an area like this to more of a native forest, right, in Timor, and to understand how big of a change in nitrogen cycling has been induced by this forage plant. And then additionally, in Sumba, right, there's huge areas of grassland in Sumba and very little data detailing the carbon dynamics of these grasslands, which I think are probably pretty unique. So we were able to look at some soils while we were there. These are two of my field assistants, my children helping me here. And you can even see a fire in the distance, right? While we were there, a large fire was sweeping through. And so I think there's a lot of opportunity to collaborate with folks in Sumba and to both train students and to gain a lot more knowledge of the grasslands in Sumba and the carbon dynamics, especially in the context of increasing drought. So with that, I'll conclude my talk, and I have been happy for your attention. So thank you very much.
Okay, thank you very much, Professor, for the inspiring presentation on this is new for us on grazing and burning across grassland system and their implication for sustainability. For now, I would like to open the floor for the audience. Feel free to raise your hand and maybe use the chat box to ask your question. Okay, is there anyone want to ask? Maybe me, Professor. Hello.
Okay, I think you were gone for a second. I think you're back now.
Okay. I think this is interesting, Professor. Because I work on instrumentation. I built some like sensor to measure something and I interesting. I see that you measure C and N, Professor. This is carbon and nitrogen, right, Professor? Okay. I just want to ask, what components are measured in the soil, like N and C, and how to measure them, Professor? I mean, what is it, like a gas or maybe a chemical compound? Because maybe we can next collaborate to make some device to measure like this component to measure the grazing or fire burn, Professor.
Yes, excellent question. So the sampling technique for carbon and nitrogen in the soil is we collect the soil, we get an aggregated sample to make sure it's representative of the soil around us,
Hmm?
and we run it through a screen in order to make sure that we can homogenize it and get a representative sample. And then
Yes.
we'll grind it into a fine powder after it's been dried and weigh it into
Okay.
small tins, and we use a carbon nitrogen analyzer. So it's an elemental analyzer. Costech makes them, but it's it's the what happens is the soil sample is put inside a small piece of tin, and it's
Okay.
combusted. It's combusted in the chamber of the machine, and then the gas is where the carbon and nitrogen are measured. And so it's totally volatilized into carbon dioxide and nitrous oxide, and then those are measured by the machine, and those are the data that we get.
So you mean you burn the soil and measure the gas, like the carbon or nitrogen, like that, Professor?
Yes. Yep.
Uh, okay. Thank you, Professor.
Yes, you're welcome. Thank you. And I apologize to the audience. I know that it would be easier to ask good questions if I spoke good Indonesian. So,
Thank you, Professor. So if we make like a gas sensor and we measure directly like this nitrogen and carbon, it is possible, right, Professor?
yeah, it would. So we have to use the standard methods. And so I think I think probably using an elemental analyzer for the carbon and nitrogen is sort of a standard method. Now,
Hmm.
phosphorus is another piece that we haven't been able to measure yet, which we need to start measuring. And so phosphorus is usually measured using a colorimetric test. So we have to, so when dealing with soils, if you want to measure something, you need to extract it first. So we it's usually some kind of acid that you leach the phosphorus out of the soil, and then you add specific reagents to the leachate that comes off, and
Okay.
those reagents make the solution turn a certain color depending on how much phosphorus is present. And so the colorimetric method for phosphorus is another place where sort of improvement or changing of the methods is important. And in addition to that, there's things that I haven't even talked about that are interesting and useful to know. Things like relative humidity, understanding how wet the soils are at depth during parts of the year, because drought and dry soils can also be a big piece in how carbon will volatilize, turn into carbon dioxide through microbial processes during really dry times. And that's especially true in these soils that are clay and have these big cracks that open up during really dry times of the year. So, yeah, lots of different ways in thinking about instrumentation, and also even measuring the measuring how hot the fires get as they move across the grassland is an important factor.
Thank you, Professor. Maybe we can discuss again later, Professor.
Yes, yeah, thank you.
Is anyone else want to ask?
Jadi nanti yang satu ini tetap menggunakan satu ini ya.
Mas Ade. I'm going to ask a question to Pak John.
Yes, Miss Lela,
Okay.
please.
Yeah. Okay. I'm Leila, John. Okay. That was very interesting presentation. Anyway, I'm working on the bacteria and fungi as well. So on microbiology. So you were mentioning bacteria and fungi as an important order role. How to say? Yes, the important role. They has important role in the carbon sequestration. So I would like to ask what. You mentioned also about the fire and grazing, yeah, can affect the carbon sequestration in the soil. So how far? I mean, these two activities can affect the balance bio and fungal activity in soil with fire and grazing. So is there any significant effect among them? Thank you.
Yes, thank you, Ibu Lila. It's an excellent question. And I think the largest effect that we're seeing is how strongly grazing, especially in the absence of fire, how strongly grazing with the urine and feces coming from the cattle, that these are really high quality substrates for fungi and bacteria in the soil to utilize. And so when you're providing these really high quality substrates to the soil organisms, that they're really able to increase reproduction, metabolism, and they are building up large amounts of necromass in the soil. Again, a little deeper, about half a meter down. And so when you add fire, right? Fire volatilizes nitrogen, and then volatilizes carbon, right? So CO2, nitrous oxide. You lose a lot of nutrients when you burn, and so burning does not have the same stimulating effect on the soil microorganisms that grazing does. And then when you don't graze, right? When you don't graze, because you just have more material, you get slightly more carbon when you burn. But when you don't burn, the lack of grazing really decreases the production of bacteria and fungi in the soil. So we can say that that the soil microbes are really sensitive to the quality of substrate that's being passed to them. And right now, it looks like the urine and feces from grazers are the best, in the absence of fire, are the best way to get nutrients to the microbial community.
Okay, also all right. So the feces is much higher. I mean, in fact, on the bacterial fungi activity, the feces from the grazer, and
Yes.
is that? Oh, okay.
Yeah. Right now we can say that as long as fire is really infrequent, so as long as fire's not every few years, it's maybe once every 10 years, then you can build carbon in the soils using grazers. Now, we know this is a problem, especially, in a lot of places in Indonesia, especially in the really dry areas that have a long dry season, the grassland burns every year, right? Every single year, it burns. And so you are starving the microbes when you're burning so frequently, and you will end up mining your soil of nutrients. because your microbes are just really desperate and they're trying to find anything they can in order to metabolize. And so they're going to mine every little bit of the soil to try to continue to metabolize. So you're mining the soil and you're not driving carbon into the soil and when you're burning at such high frequencies.
Okay, I see. Okay, then as far as I know, I think the fungi maybe will more how to say persistent because they have like the spores in it. So with the very dry and maybe no nutrients, so they still can how to say growing well. But for bacteria, maybe yeah, they will stop because there is no nutrient at all. If you said that if the dry and no nutrient available for them to grow, so maybe and it's quite very how to say affect to the bacteria and fungi growing. But actually, what is the different areas? I mean, doing this experiment in Indonesia and as well as in Malaysia and in the US, so it is much better. I mean, for the carbon sequestration in Indonesia or in America.
Yeah, it's a really interesting question, and we don't know a lot about the grasslands in Indonesia,
Mm.
especially, when you get sort of an NTT, you get towards Sumba, Timor, like we don't know a lot about those grasslands.
Mm.
And so there's no real good estimates, for instance, how much carbon is stored in grasslands right now on the island of Sumba. And we also don't know what factors are really important because, as you know, like Java is really different from Sumba as far as climate.
Right, right.
And so when you have such a long dry season like you do in Sumba, what effects does almost annual drought, right? What effects does that have on the microbial communities and their abilities to sequester carbon? Are there certain times of year when those grasslands are really pushing carbon into the soils and they're sort of dormant for the rest of the year? Or, are there, again, is fire frequency really dominating in those systems? So it connects both carbon dynamics, but also livelihoods, right? Because how important grazing is for so many families. And if the dominant grazing pattern is actually mining the soils, right? Then that's something important to know so we can address that in management,
Oh,
in sort of management efforts.
right, right. I think that's a very good opportunity for us to collaborate with you. So, okay, yeah. Okay, thank you, Pak John. Terima kasih, Mas Ade.
Okay, we still have 10 minutes in this Q&A session. Is it anyone else want to ask?
Bapak Ade, I want to ask something.
Oh yeah, yes,
Good morning,
Salma.
Professor Chun. I'm Elma. I'm from architecture. So I might be some kind of outsider in this topics and discussion. However, I want to know this topic about technology. It's sometimes related with our architecture topic, which is how we can the environment. And I want to ask you about, could you elaborate on the key of the technical variables that you measure to understand these patterns? Or it's like, how can you know that it's need to be chart every 3 years, or how can you read the situation?
Yeah, so it's an excellent question, and it goes back to um, the design of the project, right? So ideally, we could measure fire frequency in every third year, every 10 years, every 20 years. And maybe in the future we will amend the experiment to be able to look at those types of frequencies. And additionally, there's so many different features of the landscape that have such a strong effect on the results. So I didn't focus on it too much in the results, but I had mentioned that here in the US, one of our systems is very sandy, and another system has a lot of clay, right? And so we find that in the clay-rich soils that we actually have much higher concentrations of microbial DNA because those soils just provide a lot more habitat, right, for these microbes to live and metabolize. Whereas the sandy soils, the pore sizes are really large, so water moves through them pretty freely. They also dry out pretty quickly, so there's less places for water to be retained in order to maintain microbial metabolism during the drier parts of the year. And so we're finding that soil, in addition to all the different treatment effects, we're finding that soil can actually be a really strong determinant. The soil type can be a really strong determinant in how these changes progress over time. So, I would imagine, and we'll have to see over the years what we are able to demonstrate, but I would imagine that those sandy soils start to hit a limit because they're sandy until they build up enough organic matter to actually change the function of the soils, right? Because just a sandy mineral soil is not very good at holding on to and providing habitat for microbes. But once that organic matter starts to really form, then you start changing the soils themselves and they start becoming better habitat for microbes. So it could be that these sandy soils will start to catch up and maybe even surpass the clay soils once they're able to build up enough organic material for the behavior and the habitat within the soils to change. So yeah, when thinking about materials, the soil itself and the mineral content and composition of the soil, especially because again, we have not yet been running these experiments for 10 years, right? And so when thinking about the mineral content of the soils, that sets the tone very early about how these systems respond. But again, in another 10 years, maybe these systems start to build their own organic matter and respond very differently. So
Okay, thank you, Professor.
Okay, thank you. I think the time is over for the session one. Thank you, Professor, and thank you for everyone for the thoughtful question. I hope this discussion will inspire further collaboration or maybe research and innovation among all in this field. Now, I would like to invite our MC to present the certificate of appreciation to Professor John Missler.
Okay, I might take a picture in 1, 2, 3. Okay, thank you so much, Dr. John. I give it back to the moderator.
thank you. Yes, it was very fun. Thank you for the invitation.
Okay, thank you, Professor.
Thank you
Okay.
very much, John. Okay, yeah.
Okay, now we enter the second session. We changed the plans. The second session will be held by Professor Qi Jiang. Professor Qi Jiang will be from Yangzhou University, China, from Jiang, work on bioenergy, biomass conversion, and ultrasonic machining. Will provide important insight into how advanced engineering can drive a sustainable energy system. So we are pleased to invite Professor Chi Zhang or Professor Sarah, right, Professor? To deliver the presentation.
Hello, can you please hear me?
Yes, Professor, we can hear you.
Oh, okay. So let me share the screen now. I'm with my team member, Professor Wei, as well. Can you please see my
Okay, Professor.
screen?
Yes, Professor, we can see
All
your
right.
screen.
All right. So I'm so grateful and thankful for Professor Lala's invitation, and I have a chance to stand here today to give a presentation about our team's work. I will briefly introduce new technologies and equipment in feed drying and processing. So it's all related about modern intelligent agricultural machinery research team. I'm from College of Mechanical Engineering, Yangzhou University. Thank you very much.
You're welcome,
This
Professor.
is my presentation outline. Will include the team profile, the key technologies and achievements, and my experience in international collaboration. Okay, first I would like to introduce our university platform. The agriculture machinery major of Yangzhou University was founded in 1957 and started enrolling undergraduate students in 1986. So far, it has accumulated over 60 years of experience in running the program and conducting scientific research. So we offer following degree programs: the doctor program of mechanical engineering, and under that we have 7 level of discipline of crops production equipment and its nomination, and the glassland machinery equipment and automation. We also offer master program in agricultural mechanization engineering and Master of Agricultural Engineering and Information Technologies. We offer bachelor's degrees in agricultural mechanization and automation. So here is our platform. Our leading professor is Professor Zhang. He is a key chief scientist in our team, and he has almost as a PI for our country national program project, and he has as a PI to develop of China's first Beidou agricultural machinery pilot with independent intellectual property rights. So under him, then I'm now is developing another related research about animal feed drying and processing equipment. So it's about me. I am the co-chair of the Asia Pacific ADC International Drying Conference member, and also conference, and also I'm the member of the key laboratory of green and intelligent feed processing equipment. And this funding was funded by Ministry of Agriculture and Rural Affairs. And also, I'm the director of the International Exchange Office of the College of Mechanical Engineering in Yangzhou University. So I got my PhD in Industrial Engineering and Manufacturing Systems in Kansas State University, and also get my bachelor degree in University of Wollongong. So I have been engaged in this research development and application of new technology and equipment for fishery and livestock, feed drying and processing for almost 20 years. So published around 40 plus paper related with design optimization and of complex engineering system, fluid machinery design, and heat mass transfer model in related research area. So let me give an introduction about the background why we conducted this research. We know that China have large population, and Chinese people they are prefer to eating high protein meat. So fish is a very good protein resources. For industry production, normally we have to develop lot dryer and extruder. Then the production rate nowadays can up to 10 ton per hour. So we produced, we manufactured the dryers, extruders, millers, coating machine, and then use this machine to provide or produce the aquafeed feed. And then we feed this feed to the fish. So during the research, some key technologies or scientific issues we have to we basically to conduct our research in 4 perspectives. The drying equipment design, including structure optimization, and also extruder die design. So this is first part related with equipment design. And second part, we will focus on quantitative research, equipment, and drying process optimization. So this second portion will normally involve like finite element model of processing environment, drying airflow distribution, and prediction model, a flow channel characterization, distribution field, and total energy quantitative evaluation. So besides of the equipment design, like multi-zone, green energy efficiency, we also have to consider using the software to study the airflow distribution and compute the optimization design. The third portion will focus on drying quality, extrusion effects, product texture, and functional property control, quality control. So quality control also involve water solubility, moisture content, drying process optimization, and process control of the process meter. And number 4, we also study the mechanism of the feed drying and process quality control. So it's related about molecular simulation. So we have to study the biological system reaction and also multi-physical field reaction and molecular reactions. So through this kind of simulation strategy, we study the insight in the microenvironment, how the molecular react with each other. Our team will focus on from mechanism to control. So we think that we have to work on the drying quality, and first we have to focus on the drying stability and drying flow unity for intelligent drying environment prediction and control. So there are so many drying process meters like dryer structure, temperature, guide vane, angle, material thickness, material type, fan frequency. And through these parameters, we are trying to study the effect of these parameters on the airflow. Because in order to save the drying efficiency, the airflow uniformity is very, very important output. If the airflow flow is very uniform, then that means every feed can be treated equally. So in order to achieve that goal, we have to consider many parameters affect the airflow uniformity through experiment and also simulation. And the final objective is we want to develop an intelligent control of drying system to control the feed's moisture content, water solubility, and nutrition quality. So here is a dryer we developed by our team. You can see that the feed was put to the conveyor, and then the feed was on the belt and followed the movement of the belt. And then the airflow was crossing the feed, so take away the moisture content of the feed. And so to see a single chamber, we can see that the airflow initially is low temperature, and then it was heated by the heater. And so the hot airflow was there and to take away the moisture content of the feed. So in order to study the complex system, we just take out one part of the dryer and then construct an experimental dryer to study the airflow better. So this is the drying experimental platform in our lab. And then we try to study the airflow inside the dryer. So I have asked my master student to study the different guide vane angles, maybe 30 degree, 40 degree, 50 degree, and then the flow strainer. So put some strainer to direct the airflow. And also, in order to study the airflow, we purchased the 9 sensor to put on the belt, put on the surface of the feed, and to test the airflow using sensor. Our team, for the past few years, we try to investigate the influence of the material layer thickness on the airflow. We developed the different material 6, like 20 millimeters, 30 millimeters, 40 millimeters. And then we put the sensor on the feed thickness on the feed layer and test the airflow speed and the air velocity. So based on that, before that, we established a simulation model using computer fluid dynamic software tools. And then this is a prediction. After that, we use an experiment to verify our simulation result. So the objective is to set up a prediction algorithm. And we also studied the influence of fan frequency on airflow. We studied different fan frequency like 20 hertz, 30 hertz, 40 hertz, 50 hertz, 25. So by studying different air frequency and experimental design, we try to catch up the effect of fan frequency on the airflow. But at the same time, we set up a simulation model. We verify our experimental model with simulation model, experimental result with simulation model. So this is a real test result, and then comparing with simulation model. Besides the fan frequency and the design structure, we also study the influence of drying parameters on airflow uniformity, develop a multi-parameter coupling simulation model. The purpose is trying to establish an airform uniformity control algorithm. So you can see from that we use different methodology to establish a real airflow environment and real flow simulation model. And the purpose is to achieve rapid screening of optimal drying parameters. Last year, our team also trying to study the multi-parameter coupling simulation model. We found that we have so much drying data for different type of the feed, okay? And we conduct so much experiments. The next question is, how can we predict the drying process using input parameters? So like L velocity, temperature, feed properties such as bulk density, particle size, they will all affect our moisture content and energy consumption. But that is a black box. We don't know how it works, but we just get so many experiments data. And then our team trying to adopt a BP model, a new AI prediction model, a physical model correction, backpropagation, neutral network model. So we combined this model with our data to predict a related prediction model for feed drying efficiency. So that is what we do. And the purpose is in the future, maybe we want to, we know this kind of material and we want to reduce the moisture content to 10%. So when we take a picture of the material and the drying system will automatically dry it, they will set up the parameter by the dryer itself and then dry the feed. When we get the feed out, it exactly meets 10% moisture content. Besides dryer, we also study and research on the twin screw extruder machine. So let me introduce what we developed, twin screw extruder. The twin screw extruder normally contains a motor, a container, extrusion pod, and a cutting device. So the pellets after dryer, before the dryer, the pellets and the powders, they have to be milled into fine particles and then extruded in extrusion machine. So this is the extrusion machine in the industry. You will see how it works when different powders and particles was going to the twin screw extruder, and then the twin screw extruder will mix them and then extrude it as a particle. So our team also focused on study a multi-physics fluid simulation model of the extruder barrier. So this study will focus on the extrusion mechanism through multi-scale analysis. It's trying to establish an intelligent optimization strategy for extrusion process to facilitate the subsequent drying process. So we know that there's so many extrusion parameters like structure, barrier, temperature, school, materials, and extra. So during the extrusion process, the extruder normally is the key equipment in feed processing, and it is very complex because it is a high pressure confined black box environment within the barrel. And so we have to study the field of the shear rate and resident time, velocity field, and viscosity field in order to better control the feed quality such as expansion rate, density degree, and gelatization degree, moisture content, water solubility. So our team has designed different twin schools in terms of a mechanical design. We design different types of the school, and then we try to using the simulation software to study what it looks or how is the velocity, how it's viscosity, and when in the extrusion process. And so this is a process optimization during extrusion. We did a lot of simulation on the left-hand side. You can see there's an impact of the school structure parameters and the configurations on flow field characteristic in the extruder barrel. And then on the right-hand side, you can we study the different extrusion process parameter and try to optimize it on the extruded feed properties such as water solubility and also density using 2 to 3 factorial design or response surface design, experimental design. And also this year we are focus on the micro or minimum or super small 0.5 millimeters aquafit feed for baby fish. So to develop this kind of new product, then we have to develop a new design, the extruder. But the key component is a die. So you can see that our team designed a new die and studied the different design, effect of different die design on the small micro feed quality. And we found that the die design is the key element to control the size of the aquafeed size. And we also studied the impact of the structural optimization. So we compare the before and after, what is the structure design on the extruder feed property. So besides that, we also studied the material composition, molecular reaction for single extrusion and twin extrusion. So doing the different design and different feed rate, the molecular reaction will be different. Okay, here is an example of our last year our team research. We found that different school speed will finally affect the feed quality. And this feed, because this feed have to be thrown into the water for fish, crab, and the shrimp. So we found that by changing the school speed, it will reduce the water pollution because it will have the significant impact on the feed quality. And so Professor Leila and Alma has visited our Yangzhou University, and they know that our university collaborates with local extruder supplier, Fengsang Company. They are world's second largest feed processing equipment provider. So this is an experimental platform in our university our students used, and we work with company engineering. So students had the chance to see the real industrial platform. And the company also has a very has a globalization organization, and because they have supplied over 140 they have supplied extruder and dryers to over 140 countries. So projects are set up in Indonesia as well. How many minutes? Oh, 5 more minutes. Great. And here is some international student internship. When international students from One Belt and Road countries, especially from Pakistan and Bangladesh, India, So we bring them to visit company, and they have the chance to get the student internship, so they see the real product of the industry. So this is some activities. Okay, like I have experience in international collaboration since I in charge of our college international affairs, and we offer educational platform for overseas students. So we have over 140 international students in Yangzhou University. Around 80 students are in undergraduate program, and another 70 students or 60 students are pursuing master and a PhD program. Let me please allow me to explain about our undergraduate program. We now not recruiting undergraduate program, or we are no longer offer undergraduate program for international students, but we are increasing to recruit students in master and PhD program. We welcome students to here study the PhD program. And also, our government offers scholarship. If students want to study master or PhD in China, for master student, the government offers 30,000 RMB per year to support the student, and also offer accommodation. For PhD program, the government offer 40,000 RMB per year. I think that money is sufficient to cover the tuition expenses and also the living expenses in China. So that is a very good opportunity for students from Indonesia and who want to further study in China. And we are trying to develop an international student platform resources with the company and also a variety of industrial area. And I also have 30 plus years of overseas study and working experience. So I can understand and respect the students from different countries and also respect their culture. So I'm very honored today here to give a presentation because we and Professor Leila collaborate for very long time. For during the past 1 year, both of us have published 2 papers, and also it's very glad for me to invite Professor Leila, Emma, and the officers from your university to visit Yangzhou University. I think that China are very interested in globalization collaboration, and Indonesia's strength is they have their unique location and their ingredient materials, and we are looking forward in-depth research and collaboration in the future. So we hope that we together can develop a food or feed process technology in the future. Okay, due to the climate change and there are several impacts, global food and water security is widely recognized as the most pressing crisis that pose a severe threat to national stability and human being worldwide. So in the future, we want to promote a sustainable, greener collaboration with all One Belt country and also India, Russia, and many other countries, so they can join together to develop a better research. The last thing is that I will be the guest editor for Food Journal. And we have a special issue. The impact factor of the journal is 5.1. It's a Q1 journal. So the special issue topic is recent advancement in food processing technology for enhancing food quality. So me and Professor Wu Ming are engaged in this special issue, and we are welcome the related paper submission for this special issue. So we can also give a special discount. All right, so thanks very much, and I'm so appreciate for the invitation. It's just 30 minutes. If you guys are interested in future collaboration, you can add our team's WeChat, Qi Zhang, and my team member, Professor Wei Wenguang. Thank you so much.
Okay, thank you, Professor. I think this is an interesting presentation about your design, optimize the drying system. Now I would like to open the floor to the audience. Feel free to raise your hand or use the chat to ask your question.
Can I, Mr. Ade?
Yes, Ms. Amel, please.
Good morning, Prof. Sarah. I'm very interested for your presentation about the storage of it drying. And I'm interested in the heat exchanger that you use in your machine. What kind of the heat transfer heat exchanger that you use, and what kind of the thickness of this heat exchanger? Maybe it can be effects in your machine or no. And secondly, about the how to make sure that your feed that drying is good enough in the protein and never lack of the nutrition. Thank you very much. So two questions. Yeah.
The first question is regarding what kind of exchange heat exchange do I use, right?
Yes, the exchanger you use in your machine. Yeah.
And how about your second question?
The second question, how you can make sure that your food that it is drying is lack in the nutrition or in the protein content.
Okay. So how to make sure the fit nutrition, right?
Yes.
All right. Regarding the first question, we know that the heat exchange, we have the heaters supplier in China, and based on their drying production capability, maybe up to 10 tons, maybe up to 7 tons, 6 tons. So different production capability or production volume. We have to calculate the airflow and then decide which kind of heat exchanger machine we will use, and then we will order from our supplier. So there is several suppliers, there's several different heater exchanger. So it's depend on the production rate. And regarding the second question, how do we make sure the feed nutrition? Actually, we have to control the drying time and extrusion time. During the industrial processing time, the extrusion time, the maximum is 30 minutes, and drying time is normally 30 minutes. So the temperature, the very high temperature sometimes will control within 130 degrees Celsius. And we make sure that the suitable processing time and drying time extrusion parameters will not will not destroy the feed nutrition. And then we get the pellets. We will analyze the composition. And before we put in the extruder, put in the drying, we will analyze how much is the protein, how much is the fat, how much is the starch. We will do analysis, and after that, we will do analysis the similar comparison. So based on the 20 years industry experience, so the suitable processing parameter will not affect the feed nutrition. And then following experiment will be conducted by the animal test. So we give this feed to feed the fish, and then we cut them, and then we check and they getting weight. So make sure they are healthy. So to this process, we make sure the process parameter is all right. Hopefully I answer your question.
Yes, it's an answer. Thank you so much.
Thank you.
Okay, is anyone else want to ask? Halo, adik-adik mahasiswa, ada yang mau nanya nggak? Okay, maybe me, Professor. Professor, you mentioned that airflow is very important in the drying system. Am I correct, Professor?
Yes.
Yeah. How do you control or maintain the airflow to ensure it remains uniform through the drying system, Professor? Could you please explain how to measure the airflow? I mean, using what kind of sensor maybe, and what the velocity of the airflow is considered the most effective in this process, Professor?
Oh, all right. Just I explained, the feed was layered on the belt, right? There's so many feet, and in order to make the airflow uniform, first step is we have to set up a modeling and trying to simulate the airflow. So different design will obviously have the different impact on the airflow. And normally in the belt, the central one, the air velocity is high, and the two sides is normally has very low air velocity. So in order to change that result, so if this kind of bad design, so finally it will cause the feed. So in the feed in the between, sometimes it's dry enough, and the feed at both sides is still wet. So what we do is, this is an unavoidable problem. So we try to change the different mechanical design, like the band, the different convex size, so to try to make the airflow evenly in the dryer, and we put the sensor on the surface, and the sensor is from Siemens. We have published a related research paper in drying technology, and then to try to catch the airflow velocity on the surface. But only doing this one is not good enough. It still will cause uneven distribution. So finally, we change our design. It's like we have a multiple different zone of dryer. So this dryer maybe is this way, and then next dryer is this way, and next dryer is this way. So by connecting different dryers together, and then finally it will significantly reduce the uneven distribution of airflow. And then we take the samples when we take the samples from different position of the dryer belt, different position of the belt, and took out and test moisture content, we found that this kind of design will significantly change our final product quality. So the dryer pellets will become very even distributed. The moisture content is very distributed. It's very even. So just now I only explained one dryer chamber, but actually in the whole dryer design we have different chambers to tie them together. That will eventually reduce the airflow distribution problem.
Oh, okay, thank you, Professor. So we use multiple dryer instead of using one dryer, Professor. Like that, Professor?
Yes. And also we're trying to optimize one dryer chamber. So and then by changing different structure.
Okay, thank you, Professor. Is there anything anyone else want to ask?
I would like to ask, my name is Vianti. Can I?
Yes, please.
Good morning, Professor Sarah. My name is Vianti from Universitas Negeri Semarang. I joined your speech, and then I recognize that our research is a little different, but I listen about your promotion vacancy of students for master degree or PhD. And how about if we have some collaboration in writing manuscript? Can we do some collaboration writing a manuscript? For your term of heating and your research is about drying, but in my research, heating treatment in here is for structural formation. My research about thin film in solar cell. Even this is a little different, but we do some heat treatment. So in here, I would like some suggestion about manuscript collaboration. Is it possible, Professor Sarah?
Yeah. It will be my pleasure. Actually, our university are also and a company are trying to explore more resources like in Indonesia, in German. We have institution in German, Denmark, and also USA. So this is a globalization institution. So we want to recruit or welcome professors who are interested in this area to join. And we also want to establish a long-term collaboration relationship, not maybe just for 1 or 2 years, maybe it's a long-term. So I think in the future, both parties can have the opportunity to discuss and work on the collaboration together. Last year, I'm very glad Professor Leila invited me to visit the university, and we have a talk, and she also gave me some local starch. And Professor Wei he's working with me, and we are trying to work on the viscosity of these starches together. So I think in the future, we have published 2 papers with Professor Leila. One journal, one Chinese, and we are still writing on the 3rd one about going to submit to the food of journal paper. So we are working on this direction, and we welcome the faculty to join us. It's really welcome.
Thank you very much. Okay, thank you.
No problem.
Okay, Professor, we have one question in the chat box. I will read it. In freeze drying, we usually add sucrose as a protective agent for the material. For your heat drying method applied to food product, is there any specific pre-treatment needed before placing the sample into the dryer? to maintain their stability and the quality, Professor.
Okay. Let me read. Give me 30 seconds to read. Heat drying apply for then a special pre-treatment before placing. Okay. Freeze drying is normally used in the food area, especially the fruit, because the hot drying will change the color of the vegetable and the food. So the color is not nice and cannot be sold in the market, but freeze drying is very good drying method to avoid this disadvantage. So the freeze drying normally they have to put a protective agent, and sometimes it's a binder, sometimes it's the other kind of material to prevent. But nowadays, I just attended the ADC, the Asia Pacific Drying Conference last week. So nowadays, freeze drying, they also maybe not need to put a protective agent to reduce the cost. But for heat drying method, for our method, we don't have a specific pre-treatment. So the pre-treatment, the whole process is like that. First is the raw material, and then we have to do size reduction, and then extrude, milling process first, and then extrusion, then drying process, then finally coating process. So if you think about special pre-treatment for the drying, no. But if you see special pre-treatment during the extrusion, yes. Steam, water, and the feed rate has to be adjusted. So if you can see this is a pre-treatment to drying, yes. But it's just talk about drying process because we already take the feed out of the extruder. Then it will just directly from will directly flow into the dryers and dry directly. So there's no pre-treatment. And in the real industrial case, sometimes is we have a 6-level food floor. We have so much high floor. So the material is first from the 6th floor and then flow the muted and to the 5th floor. The 5th floor, and then the material will flow from the 5th floor and drop into the extruder machine in the 4th floor and extrude it. And then extruded, then going to the number 3 floor, then drying machine. And after drying, then jump to the 1st floor coating. So this is a whole process. So if you just talk about the drying process, there's no pre-treatment. If you talk about the whole processing, yes, there is a pre-treatment in extrusion stage. And extrusion process is even complicated. It may need a conditional to do the pre-mixing. Yeah. Hopefully I answer your question. No, welcome. Thanks, Mr. Kaya Singh Plapi, to ask this question. Thank you.
Okay, thank you, Professor. Is anyone else want to ask? We still have 10 minutes for this Q&A session. Or maybe me again, Professor. Could you elaborate on the efficiency on your system, Professor, regarding energy consumption and operational cost, Professor? How significant the improvement compared to the traditional drying technology, Professor?
No problem. You are welcome. I promised Professor Leila to give her seminar last year. I just made my promise come true.
Thank you very much. So maybe I think Ade has a question. Yeah, last question. Oh yes, please.
Hello. Yes, Professor. Can you please elaborate the efficiency of your system regarding energy consumption and operational cost, maybe? How significant the improvement compared to the traditional system, Professor?
When we initially developed this dryer with the company, actually they don't have such product. They have a very old dryer, and the drying process is considered during the whole production line. The dryer is consumed 80% of the drying energy, 80% of the energy consumption, and that is very important to develop a new dryer to improve the drying efficiency, as you mentioned. So so we have no clue, and then we invited the expert from the USA, and then to teach us what is the more advanced dryers. So we come on the first generation product is a Merriger, and we sell these dryers to the worldwide because two unique characteristics. First is airflow is very even and very stable. It can dry the product. The product MC variance, moisture content variance, can control within 0.75%. Sometimes it's 0.5%. So it's much higher than other related to feed dryers. So it's because this very uniformity characteristic. And also the second thing is very large volume. We have different capacity, up to 10 tons per hour. You know, so because of the design and the energy efficiency, comparing with other dryer, it's probably the you know, in order to achieve the very uniform airflow, then it will automatically reduce energy consumption. So we promote these dryers, and then we sell to many our customers. So it's around like 0.5 billion business. So be sold within 1 year. And after that, we think that for second generation, this is first generation, and second generation dryer is now we think that to reduce the heat energy efficiency is trying to have a heat recovery. But how to have a heat recovery? I have mentioned that we have different zone, we have different time, we have different zone, dryer zone, right? So the first zone maybe is 100 degrees Celsius, second zone 120, and third zone maybe 17 degrees Celsius temperature is enough. So based on the drying curve. So what we do is that we have taking the hot air from the dirt zone, and then recover and supply back to the first zone. So this kind of drying recycling will significantly reduce the energy efficiency because the heated air can be recovered. And the third generation now we are trying to do is AI. That means, just I explained, if I tell the dryer I want 10% moisture content, and then the dryer recognize, oh, okay, this is a feed for yellowfish kind. I know this kind of material, and it will automatically start up. It will make another thing is it will automatically wake up. If there's no feed, the dryer will sleep. If there is a feed coming, then the dryer will wake up. So we already achieved that function. It's already become true. So this is one way to save the energy. And then the third generation we are trying to do is we want the dryer recognize this kind of material and it's automatically dried. So no, nowadays is still people hit the button to control, but the next generation is we know that you want to dry this kind of material, and then we just click the button and the dryer automatically dries. So this is a third generation concept. So every generation is trying to have some new idea to develop new advanced product. I hope that answer your question.
Yes, Professor. Thank you, Professor. Is anyone else want to ask? Maybe one more question. No one? Okay, maybe that's all, Professor. Thank you. And thank you for everyone for the thoughtful question. I hope this discussion will inspire further collaboration, research, and innovation among all in this field. Now I would like to invite the MC to present the certificate to Prof. Sarah.
Will Professor Lela email me the certification?
Oh, sure. Yes, Professor, I will send it to you. Okay, right.
Okay, thank you, Professor Lela.
Thank
Okay,
you.
I might take the picture in 1, 2, 3. Okay, thank you so much.
Thank you.
Professor.
Thank you. See you.
Thank you so much, Prof. Sarah. So, yeah, I hope your daughter is okay. No anything's how to say.
Yeah,
serious?
yeah. That's why I have to rush home now. Thank you very much for invitation, and looking for future collaboration.
All right. My pleasure. I'm so happy to do that with you. Thank you very much.
Yeah.
And Prof. Wei, thank you as well.
Bye.
Finally, you can join by Zoom. Okay, yeah, bye.
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 Okay. This is the final section. Our final session will be delivered by our friend, Dr. Asih Melati, Dr. Amel. She is associate professor from UIN Sunan Kalijaga, Department of Physics, Indonesia. Her research focus on innovative nanomaterial and composite material that contribute to sustainable technology and environmental solution. Okay, this is the CV. So we are pleased to invite Dr. Amel to deliver the presentation. Miss Amel, please.
Thank you very much for your session. For the committee of the International Joint Discussion, my name is Asih Melati. I am honored to become a speaker parallel with the Prof. John Miser and also Prof. Sarah. Actually, I just a junior researcher, so I think I just present my study and my research now that not I think it's just a little bit about my activity now. Maybe not hikes technology, but we can share that I know. Okay, please, I will share my screen. Okay, I went to the global that we talk about advanced material for green technology towards green environment. We already have a talk with Prof. John Mayer, that is similar, but Prof. John Miller in the land. But for me, concern in the material that can be created and also can be developed by using physical science and also chemical science. I'm sorry, there is a sound because we must hear first, or we can continue. This outline is content of about the green technology and sustainable future, and about banana and pineapple based biocomposite and its application. So we will talk about the biomaterial, and then after that we also talk about the nanomaterial ZNO based on bajaka wood and its application. And then the last one about mineral and organic binder catalyst for thruster rocket technology. As we know that we has on the characteristic of the seminar that it must be appears of the Quran as the foundation of the life. So we have the foundation of the life in the Surah An-Nahl, that it is the meaning is with it he produced for you corn, olives, date, palm, grapes, and every kind of fruit. Verily, in this a sign for those who give talk. So in this verse, we know that there is so many natural source that can be developed, can be changed to the renewable energy, and also can be more efficiently for economic growth. Then from the green technology, what is the name of the green technology it is? The green technology is about the technology that using the natural plant, natural animal, and also natural mineral in that its content from earth. As we know that there is so many was that it is created by the earth. Actually, there is a metal about the 4% and glass about 5%. Plastic is highly 20%, and also the others is like this. But we have food and greenwash still higher than others, around 54%. That's why maybe it has become enormous numerous files, numerous source, and we can develop to another energy to become a renewable energy and also another sustainable technology. That why there is a global plastic that every year is very increased drastically. You can see in the 2024 it's around 400 million, and it's predicted in the 2060 is around 1.2 billion of the global plastic production. That's why we must, as a researcher, we must concern about the problem of the earth. And we know that waste composition matter if food greenwash is major share. This point is the losses in food system and potential for composting and also circular solution. We know that a big plastic signal persistent pollution risks and challenges to recycle. And also this one, the metal was also like that. It is have a discovery and recovery gap. Even where collection exists, the recycling and recovery is often low. So you see that there is so many was in Yogyakarta, particularly, and also Indonesia in the largely. They still have many was of the metal and also plastic. That's why our Earth is not supposed to clean, and then that's why also we know that in Indonesia some like have a highly pollution, but in the global thing we still save. I mean, from the plastic, but from the organic waste still enormous, still large. As we see, we have in this location. This location is about the green. The green is this one is less than 1 until 1.49. Then what is the effect of the green technology? It can be toward a sustainable future. On this one, because I am from a PhD student from INSA Central de Loire, France, we study about how to make the bio-based material become renewable energy, and we give the renewable such as corn and wood to become biocomposite or bioplastic or become pellet. Then after that we processing in industry. After that we can recycle and reuse, become another form. It is a bottle or some kind that plastic that can be used in daily life. After that, we can also discharge and recycle. After that, we also make it in the biomass and biosol. So we have composting. We can hold composting and also can be another product of plant again. So the spirit of green technology is how to reuse, recycle, and how to manage the biodegradable material towards the zero waste in 2050. What is the zero waste 2050? It is initially by Paris Agreement in 2015 that all of the country in Europe and another country, it is agree to have just 1.5 degrees Celsius for the increasing of the climate. So the Paris Agreement is the foundation to make the zero waste 2015. Now to work with this Paris Agreement, firstly about the cutting carbon emission, and then a combat climate and driving economic growth. In Europe or in another country, there is many kind of the development that technology comes from the biocomposite. In Europe, in example, in Dutch and also in Germany, they change the flax plant like this. Maybe not lives in our ocean. It's not lives in our region, but it is live in Europe, also in I think in Africa. Flax, there is flax, and they change flax to become a car that name a Become car. You can search in the internet that Become car is this dashboard or this body making from flax biocomposite. Then after that, we also have McLaren. We know that McLaren just announced that the seat of this car is made from glass and ham. Oh, this car is biodegradable. So if you have many car of the F1, you throw away in the field, then you can directly recycle to the earth. So it's very interesting. Not also to create a F1 car, it also create the electric motor. This is the cute Scoopy. This Scoopy also have been created from glass and hem. And then my research from banana leaves. I make a biocomposite from banana leaves, and this banana leaves will be applied to the of air frames and also dashboard of car. But this one is already testing for the dashboard car, but not ready testing for the wing of Air France Plus. So I make in this picture is about the dashboard of car. There's a challenge how to biocompensate can replace our technology. The challenge is about the mechanical and thermal properties. As you see, there is a heating and the collision of the car by another car. Automatically, it will be broken very easily. And for the thermal properties, the capabilities to attend to against the fire is very low. So for this kind of the problem, we must make a addition something or somewhat for the mechanical and thermal properties increase and the good one. And the proportion of that we can say is for to making the more strength and more have a higher and tensile and corrosion test using the SEC silica carbide for the particle adding, and for the fire, against the fire, we will use fire retardant agent, or we can say FRA. It is for making the coating in a slice of the biocomposite for banana. So, and they can be reduce the again the fire, the reduce on the thermal decomposition. So after we reduce the capability to spread more fire, we can develop another good one that it is resistant alliance with the fire and also with the combustion. In another country, we will also see that the fungi can be also make a biocomposite. That's fungi used in the wall. So they make fungi from the wall. So wall actually we against the fungi, but there is out of the box idea that the biocomposite can be protected by the element of the fungi. And another is it is a flux for making the building that it is nature building is become more friendly, environmentally friendly. So how we know that it is can be a source of biocomposite? Bamboo, alpha, corn, rami, hemp, flax, and also abaca. Abaca is some kind of the banana, but it's have a higher surface, or I think it's a larger leaves. And then sisal, kenaf is some like cassava in here, and then kapuk or kapuk, you know that it is also can be used, and also cotton and cocoa. How we can make in this one? So many researchers working on this. You can search in the literature that they develop two one. One is short fiber and number two is a long fiber. I use a banana leaf because it's included in the long fiber. That long fiber make me easy to create and also higher mechanical properties. In the next generation, you can see they can extract the cytosine from the shrimp and the cellulose from the spider and also hemicellulose or the lignin from the or another component from the snail, like that. Then how we can fabricate the biocomposite? I have two original nature from biocomposite, that is bamboo and also banana. Bamboo already live in Indonesia. It's among million years ago, and also banana. We have a banana as a tool as the third product of the commodity of Indonesia. Then after that, we wanted to coating all of them banana bamboo biocomposite using the plant rather than agent. After that, for create the mechanical properties more efficient, more highly, we use the SEC powder, silica carbide powder. After that, we make the prediction by using the kinetic analysis, and we also make sure the surface using the SAM, ADX, and also FTIR. For the F of FTIR and the effect of fire retardant, we will use cone calorimeter in the medium scale. So we have two test scale in the small scale, in the highly pore in the surface, and the medium scale in the large of the sample, like that. So firstly, banana leaf we make in the fabric collection. Fabric collection is like a kain fabric collection, and then bamboo also we make it in the fabric collection. After that, we create a matrix. Matrix is we buy from the green resin. It is resin that it is come from oleaginous oil that have on the processed via biorefinery. And then after that, we have the green liquid epoxy resin route. So there is a combination about the green epoxy resin and the fiber, natural fiber, and the name of biocomposite. So it is the commodity of the banana. Indonesia have 6%. Then the other is lower than Indonesia, except of this one. Except on the other Asia. So it is highly located in Indonesia. We have enormous of the banana leaf. Okay, for the methodology, firstly, we have a fire retardant that make a tense against the fire using the APP tape. Is from the polyphosphate, and then 1% of boric acid. After that, we mix with the green epoxy resin. After that, we have a magnetic stirrer to mix all of them homogeneously. After that, we create the instrument batch, namely is a vacuum bag resin transfer molding. First layer is the natural fiber. In this case is banana first, and after that we cover it with the vacuum bag and also apply. So the order is natural fiber first, and then peel ply, and then injection mat, and the last one is a vacuum bag. After that, we make sure that there is no air that enter the system. If we have air enter the system, it can be void. It can be make a hole. That's why it's not good. We make sure that there is no air in here. So our system, the vacuum pump and a vacuum chamber is already tested before, and we waiting until there is no air. After there is no air, we the resin through the system, through this banana leaves, peel ply, and also injection mesh. For the injection mesh is the function to make sure that that the resin is through homogeneously in the layer of the banana. Okay, this is for the original one, and use the fire detergent on it. For the increase, for enhance the mechanical properties, we adding the stick powder. And also, we already know that it is good for dust a car, and we want to have experiment how to make in the heat exchanger. So we create heat exchanger using this one. Also, that's why I am interested in the heat exchanger, because I create heat exchanger from biocomposite like that. So, and what is the different thing become this one and also heat exchanger? Heat exchanger, the main purpose of this heat exchanger, we know that there is airflow inside. So there is inlet and outlet side after we testing this one. So the testing is we using the concalorimeter testing, also using the piezoelectric sensor to make sure that there is flow is stable here, and make sure that there is no air that flow away from the pipe like that. So we have two heat exchanger. One using the stick powder, then the other is originally without anything addition. It's just addition about the detergent agent. Okay, and after that, we produce 4 paper about the biocomposite, it's bamboo and also banana. It's a good one to have a collaboration with another institution to make separate the biocomposite spirit. And then so the poster, and I also have a conference in Norwegia and also in Turkey. Then, but after I go to Indonesia, I not lose the spirit. That is the challenges for the PhD student, that he go in the country. So I continue my research in the pineapple leaves, that it is enormous also in Indonesia, especially in Lampung. We make in fiber and become a green composite, but we have created this for the grain descent for making skateboard. And then we also try another way how to, it is a long fiber. How about the short fiber? Short fiber, we have rice husk and also Excel that we crushing using in the instrumentation in instruments back in the laboratory. Name is high energy milling. We milling of them, and we make them 3, 1 without anything. And this one is using just resin and also rice husk. And then this one is resin using rice husk and also using the Excel. So in this kind of this, we already testing in the SRF and not really finished about the mechanical test. We will see that there good news about this ratio. Okay, the another part in the technology, green technology that I wanted to study is already done by my student and I lab nanotechnology. We use bajagah wood. In the literature, you can say that the species genus is Littoralix hax. The bajagah wood, we go to the milling. After milling, we adding by zinc site, and also using zinc chloride also can be. After that, we make them in the via green synthesis method. It is be heating and also stirring by magnetic stirrer, and we fermentation about 3 days. After that, we will have this residue about the powder of nanoparticle ZNO based on the bajaka wood. Honestly, I'm glad because I think that I can't do anything in Indonesia, but Alhamdulillah can be continue some kind of this. Then for making close to the particle ZNO, we do the characterization of the same characterization and also XFD characterization. For the same characterization, we calculate using the Origin software to create that our nanoparticle is already nanoparticle. Nanoparticle is have the size 100 nanometer, and in this kind of nanoparticle ZNO that our work, we that it is have 58 nanometer. Okay, and then what is the application after we can create the nanoparticle ZNO? What is application? What is the application of this one? So we use this nanoparticle ZNO to make antibacteria that we think to two bacteria, that is Escherichia coli and also Staphylococcus aureus. Sorry. Why these two bacteria? For the Escherichia coli, because it is included in the gram negative, that the blue one is the acid. This is highly, and then the another bacterium that we use is Staphylococcus aureus, that it have a gram positive. How to know that our nanoparticle ZNO is effective? Our have 3 kind of the Dix diffusion tests. We make agar. We grow the bacteria in the agar. This is name is control positive. Sorry, control negative. Control negative is the mean. Control negative is the condition of the agar system fulfill with the bacteria. Then we also use the control positive. What is the control positive? Control positive is the fulfill with bacteria. We insert the amoxicillin. So amoxicillin is highly antibiotic, and we testing our ZNO particle. We have unlike the inhibition road. You can see this is our Dix paper. See, there is a different color of zone. It means the nanoparticle ZNO is have effectively inhibits the growth of bacteria. For the diameter resistant, we have for the using the amoxicillin is 36 millimeter, and for using the ZNO particle about 20 millimeter. How about for the Staphylococcus aureus? For the Staphylococcus aureus, there is the same method, but in this place we have different bacteria that grow in agar. So we have also the higher for the inhibit zone for ZNO against Staphylococcus aureus. It has a 25 millimeter. And then what the other application of nanoparticle ZNO? As we see that our river is already wasted, already polluted by industrial works. In example, there is so many dye that content is on our river. In example, there is red, a blue, and a violet. We are concerned about the violet dye because maybe the violet is the favorite girl one to make a cloth or make another batik. So we that crystal violet maybe is interesting to be researched, to be observed. We make the crystal violet dye using the concentration. The concentration is about 2%, and we make a batch method. We give all of them of particle, and we arrange the different of time. One is about 0. It means there is no particle inside. Then after that is 30 minutes and half hour. We waiting there is effectivities or no. And then other is 1 hour. That's our 1 hour. Surprisingly, we have on the gradient is higher. So the increase of the absorbance, it means the nanoparticle can absorb on this one, or observe crystal violet using the photodegradation using the ultraviolet lamp. This experiment, actually this equipment is designed by our student before, by Dr. Widayanti. We use it together to create how to make a photo degradation can be observed like that. So we can know that there is observation. Okay, now we go to this case. I have Hibah. I have the research prize from LPPM about the thruster catalyst on red. So we create some collaboration with my friend. Actually, my friend is and have the same institution in PhD institution in South Central Valdelohar. So we make a collaboration. We make a collaboration also with our student Ivan, maybe attendance also in this event. This one, we know that there is some kind issues about the rocket and also the defense ministry. Defense ministry, there is no reduce of the monetary, so maybe it can be good if we have a collaboration like that. So we concern about the thruster on catalyst. So it is a propellant that in the inside is a pellet that the ornament of the rocket is like this. So there is my role and my student role in this place. So we make a pellet catalyst. We make a pellet catalyst from this place. You know, is two kind that want to use. The two kind is aluminum, made from aluminum oxide, and also manganese, and using the bentonite. So this one, my student created in Brin Unit Serpong, unit rocket in Bogor. or Tagalog, Tagakong. And then another is we make it in here using aluminum, mangan, and starch. What is the purpose we make in this one? We make this one for pellet catalyst in this propellant, so we can there is can be more faster to spread the position on the to accelerate the velocity of the rocket. And then the last one about the conclusion, we have the nature. That nature it is offer a fast ray, a source that can be harnessed and transformed into advanced material, offering great potential for a green technology. And another one is about the downstreaming of research in Asia can be optimistically achieved if it is supported by a research agency and industry, as well as educating with funding. Because without funding, we cannot do anything. I think you agree with me. And then the last one, the limitation in equipment can be addressed through inter-agency collaboration, inter-university collaboration also. However, laziness and pessimism will underestimate everything. Okay, thank you very much. I happy to hear the question from you. I think it's enough for me, Mas Ade.
Okay, thank you, Miss Amel, for the interesting presentation about biocomposite from banana leaf and bamboo, and you also successful produce ZNO using bajaka wood. Maybe later I ask for a small sample to try making it maybe gas sensor, Professor. And also you successful made igniter for the rocket.
Yeah.
Okay. Now I would like to open the floor to the audience. Please open the mic, or use the chat box to ask your question.
In the chat, I think there is.
Yes, we have one question in the chat. I would like to ask both during processing and long-term storage in this work with the biocomposite made from banana leaf, what kind of treatment or approach have you found helpful to prevent fungi or other microorganisms from degrading or the material? And is this research method is already applicable in industrial or still developed in the lab?
Okay, thank you for the question, Sister Kira Singgih Palupi. It's a nice question. How to against with the fungi and other microorganisms. Actually, our biocomposite is created from two main components. First one is fiber, natural fiber, and the second one is the matrix. So the matrix used is the resin. So the resin is some like a plastic, but it's come from the bio base. So don't be misunderstood, misconception about this kind of the resin. The resin actually make us to have a higher preventive of fungi and also microorganisms. So in Indonesia, there is also resin that it is that developed from the cassava, singkong, named bioplastic. This one, bioplastic. Bioplastic is the form is like layered. So like this one, layered like this one. But in my case, we use the resin using the liquid one. So it is just different form. So you can make plastic become liquid from the natural also. So how about the properties? How about the properties? The properties is also able to prevent from the fungi and also microorganism, same as the plastic. But for life cycle assessment, or we can say LCA, it is all different because we are from the composite. Life cycle assessment, or LCA, is smaller than product of the product. I mean, commercial without from natural product. But if we regard about the healthy earth, we must concern that it is no problem about that, but we can change with another, because you know that if we create some you can see laptop or have handphone or a car, like just 5 years after that, maybe you will boring, and then you will buy again. So it's not like some favorite one to become to buy a new one with a cheaper one, and also not have it much gasoline. Why it was not make a need gasoline? Because it is lightweight, so jadi sangat ringan. We have on the car is a very lightweight, and also if there is a light network, it means if you are go from Jogja to Solo, it needs around 5 liter. Using the biocomposite, you just need 2 liter. So it's very economics. In the eye of economic is good one, because you can economize the money to buy the gasoline. Okay, and then other question is, if you don't mind sharing, what were the main challenges you faced while trying to improve the durability to the banana leaf biocomposite? The durability of the banana leaf cycle in the industry meaning is LCR. Yes, it is still have challenges, because we did not know there's so many species in banana. You see that banana it is ada pisang kepok, pisang raja, pisang lain. It's automatically there is a different of the content hemicellulose and cellulose, lignin, and also lignin, hemicellulose, and cellulose. So because of there is a different of thing of this one, so we didn't know, we didn't test about the life cycle assessment. I think like that, I wonder that you understand and you can accept that my explanation, Miss Kira.
Okay, thank you, Miss Amel. Is anyone else who want to ask?
May I ask Pas Ade?
Okay, Miss
Yeah.
Nala, yes, please.
Yes. Okay, thank you very much, Dr. Amel. So that was very interesting actually. So biocomposite is quite very familiar, yeah, in biology program, I think, because it's bio-based. One question for me. So because you were doing the antibiotics or antibacterial activity, so since my background is in the microbiology, I have to ask, why did you choose those two species of the bacteria, Staphylococcus and Escherichia coli, Dr. Amel? Thank you.
I think this one is learning for Prof. Leila. Yes, I actually that's a new one for me to test in the active bacteria directly in your lab. Actually, we did it in your lab. We choose this one, two bacteria from Escherichia coli and Staphylococcus aureus. Is much of the researcher using these two bacteria to test their capabilities of their nanomaterial. So the reason one is like that. The reason two is about the price. The price of the Escherichia coli and Staphylococcus is cheaper than another bacteria. Do you agree with me, Prof. Leila?
So not really.
Yeah.
But anyway, it's very, very good. The result was
Yeah.
But okay, another question, may I, yeah, Mas Ade? Okay, thank you much.
Yes, yes.
The answer. So it is the because since you are substrate, I mean you were how to say creating a biocomposite from the banana leaves. I mean the material with the high content of the cellulose, yeah, or hemicellulose, or you know the lignin.
Ini ada acaranya IO.
How did you convince us that those material is will be easier to be degraded by fungi, because the fungi is quite very easy to degrade them with high content of the cellulose to make, I mean to create the biocomposite. I think you have you think you have also that those material have to be how to say prevented from the fungi degradation.
Yeah. Thank you, Miss Ella. If I just use the bioresin and also biocomposite, of course it is fast. So the test is I testing about the thermal properties using the TGA, thermal gravimetry analysis. It's take me for one biocomposite without coating with phyretidine, it needs around 2620 degrees Celsius to degrade in the high condition, in the high temperature. After coating it with and mix it with the fire retardant agent, the content of fire retardant agent is APP type polyphosphate. So polyphosphate can against from fire. There is function to against the fungi and also microorganism. So after we are testing in the TGA machine, it is increased from 650 degrees Celsius become 750 degrees Celsius. From the thermal purposes is higher, and the capability against the fungi and also microorganism is higher. But it is just in the theoretical theoretical studies. I think we did not ready yet to testing how much and how and the life cycle assessment for this one. If we applied fungi in the surface, we didn't test them. We just consider that IPP take have the polyphosphate that it's can be against the fungi and also microorganisms. But yes, we didn't test it yet about the activity of the fungi. Maybe we can share and we can collaboration.
Sure.
Yeah, how to make a test of the resilience about to fungi and also microorganisms.
Okay, so US now still in the developing in the lab scale. I mean, no industrial scale yet, yeah.
For in France, banana and bamboo they used to racing car in France, so already adopted this invention already adapted to make the dashboard of the not F1. I don't remember what is it, but racing car that is produced by France, because France is produce a car and another mark of car, Peugeot, Renault, and so many. So they adopt this technology. But in Indonesia, but I researched about the pineapple and also raisins. I think so. It is can be industrial level, because you know in Indonesian very hard to say that we have a collaboration industry to make this applied in the higher scale, not scale.
In the industry
Like
level,
that.
yeah, big industry level. All right, thank you
Yes.
very much. Yeah, thank you very much, Dr. Amel. Okay, thank you.
Okay, thank you. Is anyone else want to ask? Maybe me, Miss Amel. I remember when my professor in IPB University, she has successfully developed a helmet made from coconut fiber, and she successfully sold this product. And after that, he got a new car. What I want to ask is, your experiment is using banana leaf and coconut, right, Miss Amel? What
For
is
in France, yes, banana and also bamboo. But in Indonesia, I changed to pineapple and also rice husk. Because
okay?
it is yeah, it's different.
What I want to ask is, what is the basic for the selecting this material?
Okay, it's
And how far your research progress toward practical in real world application maybe, and how its performance compare with conventional material?
okay. Thank you, Mas Ade, for the question. For now, as I told you that I concern another level of biocomposite, banana and bamboo in the last in my doctoral study. Now is my second year. I'm here. I'm Indonesia. Actually, I wanted to make a different kind of the natural fiber. I told you is this pineapple and also rice husk. Pineapple and rice husk. Why I choose it? Because one is pineapple. Pineapple we have long leaves, very long leaves, and for higher mechanical properties than banana. In my opinion, like that. And what kind of another is banana have around 60 until 70% of hemicellulose, but from the pineapple it has higher than banana. It's mean if this have higher than banana, it have higher mechanical properties than banana. In my opinion, like that. But we didn't yet to make a characterization. We just characterization by XRF and also SAM, but not yet in the mechanical part. In my plan, in December and on January, we will make a test in the UEE because we have on the machine. Our lab didn't have it a machine, so we will test of this two material, pineapple fiber and also rice husk and Excel fiber to another lab. From the rice husk and also Excel, why I use it? From the rice husk, because we are agriculture country, that in Sukoharjo, Klaten, so many field it is have a plant of the rice, and after rice is harvested, become the wash of the rice husk. So a very enormous amount. If you go to the selepan, what is the selepan in Indonesia? In the finery machine, you can there is so many rice husk that it is not be used anything. Just for we use rice husk just for make the wall, batu bata, or in the stone wall, to make to combine the clay become batu bata. Another benefit of the rice husk that we know is to make another is to make gerabah, and fast and another properties that it is be sell in decorative pattern. Just two of them. Then from the technology, we didn't know that, and we didn't know that the benefit of this RISAS. So I brave to propose this two kind of material and make them become more valuable, like that, Mas Ade.
Okay, thank you, Miss Amel. Is anyone else want to ask maybe one more question? No. Okay, maybe we close Miss Amel, Dr. Amel.
Okay, yeah,
Thank you
thank
again,
you.
Dr. Amel, and
Thank you so
thanks to
much.
all participants for your active engagement. We truly appreciate your contribution to today's seminar. Now I would like to invite the MC to present the certificate of appreciation to Dr. Amel.
Okay, I might take the picture in 1, 2, 3. Okay, thank you so much, Dr. Asi.
Okay, on behalf of the conference committee, I would like to express our deepest appreciation to our speaker and to everyone here for making this session meaningful and interactive. We hope that today's discussion will inspire further collaboration, research, and innovation among all of you as future professionals in this field. With that, we conclude this session. Thank you again, and I wish you productive continuation of the conference. Okay, thank you.
Thank you.
Okay. Thank you so much to our moderator and all speakers for such an insightful discussion. Hopefully, today's exchange of ideas will inspire us to continue learning, exploring, and innovating towards a greener and more sustainable future. Before we close, we would like to invite all the participants to do the documentation session. For all the participants, you may open your camera. And Miss Dea, please help me to documentation it. Okay.
Okay, I will take a picture. 1, 2, 3. Maybe once again. 1, 2, 3.
Okay. Thank you so much, Miss Dea. And thank you so much for all of the participants and the speakers. We would like to invite all the participants to stay connected with the international office, as we will continue to hold more insightful, collaborative, and international academic activities. We will look forward to welcoming you again at our future programs. Once again, thank you so much. And wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you.
Thank you to everyone.
Thank you. Yeah. All.